Uh, Good morning, colleagues. I welcome you to day three of our SAAB 2022 47th conference. It's day three of the 4-day events. Uh, I hope you had a restful night and you are ready for the day today. To start with, I have a short announcement that I want to make before we open our proceedings. I've just been informed that anyone who needs a Sagnas, some Sagnas points must please send their names through to Medellin Struik, and she will inform them on the. She will actually forward the names to the sub secretariat to attend to those requests so that those points can be earned by those who have requested for them. So if you need points for Sagnas, just forward your name to Medellin. So to start with, we are going to have our plenary lecture from Dr. Matt Bias, who is currently based at the New Zealand Forest Research Institute in New Zealand. So I will read his CV before he can give his talk. Dr. Matt is an alumni of Stellenbosch University in South Africa, uh, undertaking a systematic vision of lobostemon, which is Baragna Shai, for his PhD thesis. Yeah, he lectured plant systematics and was the curator of the AP Kusen's Herbarium at the Northwest-University from 1998 to 2006. During this time, he developed an interest in the summer rainfall mesems, which is Isoaceae, in particular Chasmastophilium and Dilosperma. He joined the Compton Herbarium of the South African National Biodiversity in 2006, where he continued research in the Azoaceae, but this time focusing on Oscularia, a genus which is endemic to the winter rainfall area of South Africa. He also established an interest in managing occurrence and taxonomic data and initiated the trial of the botanical research and barrier management software, which is called the BRIAMS, for specimen loan administration at the Compton Herbarium and provided basic BRIAMS training and support for the Angolan Biodiversity Census pilot project. He is currently based at the New Zealand Forest Research Institute, which is also known as SHION, in Rotorua, New Zealand, where he manages disparate biological collections in an integrated manner as project leader for SHION Biocollections. Dr. Mati is also a team leader for plant systematics and collections and the curator of the National Forest Herbarium and Xylarium. His translocation to the Southern Pacific has spawned a research interest in Pacific Myrtaceae, in particular Metrosterios and Leptospermum, in response to primary industry drivers and also plant pathogen challenges which are currently facing the region. So Dr. Matt is going to give us his talk, which is entitled Being Ready When Opportunity Knocks, from DNA barcoding and honey to the phylogenomics of New Zealand Leptosperma and Kunzia. So we'll give him the floor to give us his talk. Then we have a brief question and answer session before we get into the motion of our day. Thank you very much. I now hand over to Dr. Matt to give his talk. Thank you.
Kia ora. Hello everyone. Can you hear me? And on my slide showing, I guess a thumbs up would do. Well, it's good to be here, and it's even been better to see some familiar faces amongst you over this number of days. Excuse me. Slides ready here. How do I go back? Slides. It's a funny one. Just give me. Hello, Jack.
Hi, Matt. So click on your actual presentation. There we go. There we go. Okay, and then your camera and mic is off. There we go. Okay,
We're
Matt.
back.
You're back, you're up, and it's all fine.
So yeah, thanks again. Sorry about that break. This talk does not represent a one-man show. We almost have a rugby team of colleagues and friends over the years who have contributed in one way or another. One of the three projects I'm going to talk about today. So as mentioned, Forestry Barium, which is sort of New Zealand Forest Research Institute, and in New Zealand, a lot of the research institutes also have a trade name. So our trade name referring to Scion as the Forest Research Institute in the team. I also like for primary industries who have funded almost all the work we talk about today. Technical issues on time. Right. So in New Zealand, 3-year barrier has been designated as nationally significant. And that's good news if you are, because it means the government will supply funding for you to operate. Focus on indigenous flora. The third barium, which happens to be the National Forestry Barium, has a focus on cultivated woody plants associated with forestry. The forest is quite big in New Zealand, probably the second largest earner of foreign capital after milk. And so the collections represent of the flavor of the day over the years. So we have good collection. We have good collection eucalyptus in Australia. We have a fair amount of pines collected from the States, United States, and Mexico. And more recently, we woody cultivated added to our collection. It should be woody, not indigenous, but in New Zealand. This work really does biosecurity issues. We'll talk about in a minute. Recently, we managed to survive 75 years. We had a celebration. And to this, and on the front, looking at the plant, ideas what that plant specimen, subtitle, Manuka capensis from South Africa. We have 250 in our. No idea why they were proof to be depressed with the bad weather. I go to the cabinet, draw out these specimens just to remind me where we have come from. So I mentioned biosecurity. So biosecurity in New Zealand is really a big thing. New Zealand is rather isolated in the world, and that provides us to do things other land-bound countries probably consider doing. So one of those is strict border controls into what comes into the country. pathogens out of course. And for this initiative or this effort, keeping out pathogens, the Ministry is also known as I have a unit called Biosecurity in New Zealand and protect the primary sector and the natural fauna and flora from new pests in general. So for this purpose, a large number of biosecurity officers have been appointed, and they essentially decide on walking down and just looking for pests and diseases that might be observable. So with a big data, intelligence and all modeling and all that kind of stuff, high risks identified in the country. So these put up willy-nilly. There's a lot of theory behind it. And the table below on this for 2019 to 2020. And during the course of the 7,005 were in for pests. The next cell, the next row down for the particular year, 172 samples that were sent to laboratories. One comes in, signs the contract with MPI to identify any new pest pathogens or that these biosecurity really identify in the field. A large portion of our work is then for MPI to identify new host plants. There's no from. But as things are, when these officers come across plants in the field and they happen to be sterile, they leave anyway and become identified plants, as I'm sure you will appreciate, especially in the genus Eucalyptus, for example. If you don't have buds and fruit and together at once, it's really difficult to identify some of the species. Upon my arrival, I used the opportunity not to implement a DNA identification. Free of on top of the work that we were contracted to do for MPI. So looking back, proud in saying that I think it was a good move. Number one, it sort of enabled us to streamline our DNA identification service. We made down and have a turnaround time of about 36 hours. So if the sample arrived day one, by day two, lunchtime, we would have a name. And then second, I think it sort of built up a good reputation with MBI. They sort of gained confidence in our ability to do this DNA work. Which brings me then to the myrtle rust is one of the most pathogens that have reached shore. It landed here in 2017 on this map, how this particular pathogen spread across the world. Indigenous or not, it sort of first came on the radar in Florida in 1977 and then quiet. 12, 15 years it's really exploded. From New Zealand, quite calm about things until it was detected in Australia. There's a standing joke here. Now, anything that happens in Australia, be it politics or sport, it'll blow over to New Zealand a day or two later. And true enough, first in 10 through to 2015 in Tasmania, and then this pathogen arrived. This pathogen, myrtle rust in particular, is quite unique amongst rusts in that it is not species specific. It has within the Myrtaceae family. And in New Zealand, of course, not as big as Australia, but there are genera of endemic, of which Lophamyrtus neomyrtus are endemic to New Zealand. There are of endemic species, so let me down. I think it's 28 of those 28 species, 26 to New Zealand. So on the slide, just the of examples of Tricidros species here. and have to, these species have huge cultural value, population. Examples here of Leptospermum and too closely species. Probably the closest you'll get in South Africa would be the tea tree, which is Melaleuca, so not closely related, but they look very similar. And then nwu-pukke is monotypic, only to be found in New Zealand, very non in the bush and difficult to identify. And then on the right is Opcodata and Bulata. These two species in particular have been hard hit for us. DNA work for MPI, and then it didn't take sort of a big jump of in the mind to foresee that myrtle rust was arriving in New Zealand, and it would be handy to have a DNA barcode library ready before the to assist in then the incursion to myrtle rust. So see that thought to MPI indeed, that was around about 2015. 2015, then MPI asked, for tenders to do the job, the contract. And I suspect one of the, probably two reasons, at least, that why we got the contract. Number one, we came in cheaper than the rest. Excuse me. And in addition to that, of course, we had this track record with MPI before doing DNA barcoding work for them. Of species required for the barcoding. So the target was to target every single genus, at least have one representative sequence in the library. And most of those specimens we already had in our cabinet, so for easy access to gain plant. So I think at the end of the day, 43 genera. So recall around about genera. So the rest all in cultivation, planted in gardens and parks across the countryside. And the in the of the region we targeted. So this was commercial work in essence, and of course the challenge with doing commercial work, research work, is to try and get a paper out of it. And thankfully, published in the journal Forestry. Table pictured here on the right, two regions that we initially targeted for barcoding purposes was MATK and ITS. Column from the left says the success. And if you glance at that column, you can see a lot of the accessions was 100%, and in very general, only one or a few species present. But then species numbers rise, like for eucalyptus, where we analyzed 27 species of 96 samples. For MATK, the identifying species was the percent. And then similarly for IT, a few species easier to identify. You have a lot of species in the less easy to identify. Then these weak success efficiency in MATK and ETS, we added a third region, namely ETS. And then at the end of the three regions in combination with each other enabled us, 98% of the down to either one or two species. So this was a huge success. This paper was published in 2016. We did the work in 2017. Myrtle rust arrived on our shores, and I'm happy the barcode reference library was extensively used in the beginning, the spread of across the country, to identify the host plants as the pathogen spread across New Zealand. And just about at this, like a double whammy, the next crisis to shores was honey, known as Manuka honey. I'm not sure whether this particular honey is available on your shelves in the stores. In Europe and in China in particular, Manuka honey has gained a lot of popularity over the years. Indication, what this industry is worth to New Zealand. So in the last exports to China total like $85 million. I just highlighted 3 countries from New Zealand on this. There are hundreds of from us. The total of this industry last million is 2015-16 was worldwide more money was being sold than produced in New Zealand. So that's an interesting thing there. So it turns out that these standard normal multifloral honey, placing a Manuka to earn or two, while more than an extra buck or two. Manuka honey can be 10 to 20 times more expensive than a multifloral honey. The threat to New Zealand, EU and the Chinese were threatening to stop any import of Manuka honey. The New Zealand government could guarantee that honey leaving our shores was really 100% Manuka. So there was some kind to ascertain the authenticity of Manuka honey in this country. So you can imagine it was high pressure situation from a political and financial for New Zealand. And in times like these, of course, I guess a bit of good running this is that the person government was prepared amount of money to tackle this issue. And so the barium nectar on behalf of the nectar then is supplied to laboratories who were then frantically trying to do some kind of for Manuka honey. We've never collected before, but yeah, the money was good. We signed on the dotted line and off we went, did a lot of reading. We had essentially to get this work done. I mean, time essence here, so big time. So did a search and whittled down 3 potential tracts, the nectar from the flowers, and decided upon one based on 2007. The reference is down there at the bottom of the slide. Tweaked it, but it took it pipette, sterilized water inside about 1 microliter, and then took each one by one and flushed the water, sucked it up numerous times, and in the sugars and the like water, and then these were used by other then the application technique. The top show you how dangerous it is to do field work in wildlife out. So in case you're kidding, no snakes, no, to bite you around every corner. If anything, it will be these domesticated farm animals that come and greet you. Just some useless information. The biggest indigenous vertebrate in New Zealand happens to be bats, and that's where things end. So rather safe country, but quite boring, I must say. This project enabled us to collect a lot of voucher specimens at the same time for the barium, a big bonus to us that of metaceae. We didn't just collect metaceae in this particular case, but any particular flower that bees might visit, just to ensure that the test that was developed didn't exclude, all the non-Manuka flowers. If I say Manuka honey, I forgot to mention really this Maori name for Leptospermum skipperium, of that plant. So the Manuka honey made from Leptospermum skipperium. So there's a lot of sitting behind a ute, or what you call it in South Africa, bakkie, at hand for the smaller flowers and a lot. There was time to contemplate, and it occurred to me, the government is spending all the money on doing research on honey, but we really don't know the taxonomy or systematics of Leptospermum. So just to step back a step or two, and to talk about Leptospermum in general, on this distribution range for the whole genus, so it occurs from Malaysia all the way down to South Africa, concentrated or diverse in Southeast Australia, 2 species up in 2 the rest in Australia. So, in confined to the southeast southern parts of mainland Australia, also and then of course in New Zealand. So I've never been Skippering in Australia before. I mean, I've been numerous times and walked around in the bush. James and Corey from my naturalist for evolution in the Skippering in Mania and on the right in New Zealand, born in mainland. Same, said, bit of a difference. So this is all, and I notice, the difference in leaf shape. Ovate leaves have linear leaves, the rest are more or less lanceolate. So the majority of have lanceolate leaves, but every now and then you find populations, especially down on the South Island, that have ovate leaves, and then in the bog, the damp areas, you find species then that have so there's variation there. And then when you look and flower color, even more variation. Here we have prostrate plants confined to high altitude regions, prostrate plants on serpentine soils. Those shrubs, up to about 2 meters tall. But every now and then, and especially Stewart Island, you have these stands of trees. And then as you can see, you have from really nothing new. I mean, the earliest plant collectors, European plant collectors in New Zealand, from Banks and Soliner to the Fosters and Hooker, and the botanists of Kirk, Kane, and Cheeseman, they all documented this variety. Cocaine in 1919 was sort of by all this variation and rights provided there on the top right that the forms are seemingly impossible to classify. These have been revised by Thompson in 1980, but she never had Australian New Zealand in hand. So a lot of recognized in the field names by Kirk and Cochrane in particular. In the New Zealand Skippering then synonymized all the varieties and only one. So and this is the pink flowers going to the northern bits of the North Island. Variation in the plant, and you're on an island, and what comes to mind, of course, would be radiations and islands. I mean, radiations are sort of synonymous, and New Zealand exception. So I put on here. I mean, this was I had and analyzing the honey, and we don't even know whether we're sitting in the middle of a radiation event. So New Zealand is not uncommon. Here, paper by Heidi Moit, who did some work on myosotis and the Barajan. And this is the tree. And if you have a quick look, you think you have one of those Cape clades, where you have resolution all in a short time frame. So the same in New Zealand for a lot of taxa, and this kind of course, generate traditional Sanger sequencing. Somewhere in the paper would be right. If you only had more data, this would increase the resolution. It was at this time then I attended a conference in Australia, in Canberra, the conference run by the Systematic Botany Society. And there was a post-conference workshop arranged. We learned new tricks, how to draw up species trees, and I opted for that. Lunchtime was a fair, and we opted to buy lunch at the local on the university campus. So there, with the sandwiches in the one hand and the cool other, walked, drew out a credit card to pay for the out with shock that the was only accepting cash. And me, and highly embarrassing. I had this tap on my shoulder. Behind me was Peter West, who I included the photo of, who kindly then offered to pay lunch. So there, I was indebted to him and made sure to sit next to him during lunch, if only then to get his bank would repay him. But as things, lunches, conferences, we soon started talking about statistics, genetics, and then the next, and of course, talking about the new craze, so-called NGS sequencing. Whereupon Peter asked me, Lemon Lab. Of course, I hadn't. Long story short, back home in New Zealand, went onto the internet and discovered Lemon Lab in Florida. So these guys are one of the, I guess, in the world commercialized next. For a price, they would do the for you. Quite competitive. So then we're winning as co-author papers that. So in theory, I was very happy with. And thinking of challenges that Sangha sequencing had, recent radiations and the like, I went to and said, listen here, there's this potential of doing cutting edge science. This kind of work has never been done in New Zealand before. There were no papers published that this particular technique of NGS sequencing was, there was no paper available for plants. The technique had been successfully used in invertebrates and vertebrates. There was one started work on plants just before me, but work had not been published. Nora Mitchell, that's right. She worked on the protease. She did a project on the protease. At the end of the day, we did, but then Peter, and that's how he got to know about the Lemon Lab. Long story short, I presented all the facts to MPI and told them it doesn't make sense on Manuka Honey research without having a good taxonomic foundation, at least for the individuals of Leptospermum and Skipperium. And behold, they agreed, and a month later the money was in the bank. Right, so very happy about that. But of course, that meant the work had, and here again, the time constraint. Time was of the essence. We had countries in the background, our honey, so things had to move quick. So there wasn't time to do the sampling. We tried our best to save time, to make use of all those devices that we had employed in the past to collect nectar for us as a country. We had developed that extraction technique for them. So while guys were collecting the nectar, at the same time they had collected leaf in silica and passed that on to us of material that we could select from. A slight constraint in this study that we weren't use, which samples to use. That target all the various variants that have been described in literature and targeted the like. But we had to do the best with what we had. At the end of the day, we had 27 samples and 3 Australian of the Skipperium that we had. The Australians to collect for us, mainland and on Tasmania, and then Peter Wilson, a co-author, collected a number of leptospermums to function use as outgroups. In terms of tree construction, what you do just about everything. All you expected to do when you make use of the service is you need to get hold of the material, you extract the DNA, make sure you have enough DNA, and then you make good DNA. And once you courier to them, later you get the tree box. Maybe everything goes to plan. With our time, it worked well. On Friday afternoon, I went to Auckland Airport and delivered a package to the courier down. And Monday morning time, it arrived in the lab and they took it. So in terms of these guys were there, went along with what they did, through about everything at the data. So we did a maximum analysis using XML, coalescent methods, Astral and SVD quartets, and then as well as a super network to pry the data as much as we can. In terms of stats, they designed to utilize 485 loci in the nuclear genome. So applied in across 42 samples at the end of the day. In total, then we generated about 32 million nucleons. We did 2 allele, 430,000 base pairs per allele with locus length of 800 and something base pairs. More importantly, at the end of the day, we generated 53 informative sites. So you of informative sites. To an S. So we had good to say that almost identical for so it's just focusing on the other base lineages at first, the orange clay specimen. So looking down, as I mentioned, all trees across more or less the same topology, and all branches bootstrap support. So high confidence in a range here, and us, of course, was that samples from Tasmania were immediately sister to the New Zealand. Those two then in turn and samples of leptospermum Skippering. So this is evidence that whether the land distinction in mainland and Tasmanian samples are due to vicariance or dispersal, what we can say is that the New Zealand samples most probably from Tasmania and got to distance, well, not that long, relatively long distance dispersal from Tasmania to New Zealand. So focusing a bit on the the first thing that comes a geographic signal to the data map. The colors on the dot on the map correspond to on the tree. Essentially, 3 clear groupings of specimen. The red dots in extreme, and the blue dot, the green dots, green samples in the middle of the specimens all form phyletic groups. Just about monophyletic, it's just the one oddball black WA2 cato, and then the rest of the blacks monophyletic group at all. That's the black samples didn't play along, so we went a bit, and it turns out that of all the species with the most data, most of would be then spread amongst the black samples, or the samples colored in black there. So the suspicion, the high amount of these specimens or samples had 50. Unfortunate. Perhaps the missing data then is one of the reasons, potential reasons why these play along italics and form a nice monophyletic group. But clearly to the data. So we also did a network structure. The branches leading, so all the terminal branches in the tree had 100% bootstrap support. Let's go back here. Each allele from each species then or sample then grouped, which we would expect. All have 100% bootstrap support, but then there's lower support just a little way in, and you know why we have all this data, relatively weak support every now and then. The question was that there might be some hybridization or incongruent, and so we did a network analysis behold, yes, the network analysis would have it that hybridization or admixture of genes in the past, and these events in the tree, they're sort of all towards the base of the tree or base of the network, suggesting that this process probably played a role in identifying Leptospermum in New Zealand in the past, and it has at present down the tips now, more tree-like, branch-like, rather than like in the middle. So this conforms to about the New Zealand flora development thereof in the past during the Pleistocene. A lot of glaciation events coming and going, and it's always been theorized that this has certainly played a role in other diversifying genera New Zealand. So here we have sort of evidence pointing to the same fact. And what's more, these historic occurrences probably also then partitioned the genetic data to some extent. Dots back on the map there on the left, and then indicates boundaries of the floristic centers in New Zealand as identified by Wardle in 1980. Then at least major clades, fall in one of these floristic centers. So these again thought to have occurred, thought to be defined by the glaciation events across New Zealand in the past. Economic implications of this. So there's MIM supported distinct Australian, Tasmania. Good result. We couldn't definitely say the samples grown in Australia represent different species. The type specimen Zealand. So our in Leptos was not sufficient to confidently say that those samples from Australia represent species. So the verdict is still on that one, but at least we know the New Zealand specimens species is certainly Tasmania rather to the mainland. All this data to available to us, we didn't get resolution every now and then, and the question, I guess, could be asked: do we data? Probably not. I would be surprised if any data picture. Perhaps if we add some chloroplast data to this, who knows? Time will tell, I guess. So this genetic structure reflects geographical rather than morphological considerations. So we had a good look at these groupings across the country and any morphological to support them. So it seems to be more, I think, than obvious morphological partitioning in. So it would appear of this that the relationships within New Zealand Leptospermum Skippering sort of at the true phyletics and population are things going forward, at least in New Zealand. I would advise that a good and proper study is initiated, and that then put it to rest. Yes, I'm skeptic that there are free taxa in New Zealand. Paper, and we're very proud of that. This was published in 2019. We gave ourselves a pat back, and a week later, our CEO come to his office. I kind of was quite red. So yes, I landed on the red carpet. Why? Ask. Out that the Council of Maori Chiefs weren't too happy with me. Here in New Zealand, because you have many chiefs. You have a large number of North Island, each with their own chief, and each has different clans and the like. I'm really upset with that. Gone ahead and published molecular on an indigenous genus. I was this being a government, that the government had gone ahead and got emissions in place before the work had started. Turns out they didn't. So yeah, stopped with me, and I accepted responsibility and obviously apologized. So lesson learned. That was a nice position. And this has nothing to do with the Nagoya Protocol. In fact, it has been. So New Zealand, 1940, well, to start the 20s, around about there, the Maoris in particular, was King George IV, was King of England at that time, to ask for his protection, that the French might annex New Zealand. So the British to claim another bit of the country as sovereign, not to the Queen or King, and then drew up the treaty essentially overnight, written first in English, and then hastily translated into Maori. And as they say, the devil. It turns out that as part of this translation exercise, some meaning lost or added to the treaty. It wasn't intended to begin with. So on the there was this statement saying that the Maoris retain of their property. Now, if you say property, comes to mind probably land and worldly goods, like a car and a house and the like. This was translated to Maori that the Maoris would own full ownership of their treasures. Now, treasures has a meaning to the Maoris than property. Worldview, anything that's indigenous to New Zealand, fauna, flora, and the sea, those are all cultural treasures. This from the onset, a misunderstanding taking place, and science seen ever since. In essence, ownership, and anything with and fauna. That protested, I'm sure, fell on deaf ears when Waitangi Tribunal was up to here now to help and assist settle these historical grievances. So the Maoris, they can lodge claims to the tribunal, the big investigation takes place, and then I think that the findings of the tribunal are set in law, but it's fine. All the claims have been resolved so far have the government and the Maori have decided to follow the findings, except claim in this country known as Y262. So the Y is abbreviation for Waitangi. The name Waitangi comes when the treaty was signed. That was the name of the little town. 62 states the following: that the Maori to make or participate in decisions affecting indigenous flora and fauna, and there are a whole number of other rights that they claim, and that is the first one where they claim the right decisions about the propagation, development, transport, and study, and other things of indigenous fauna. So out of claims, this is the one that has not been resolved yet. I mean, this claim was 21, and we are almost 30. There has still been no solution. Contentious. It's place. But doing this here in New Zealand, I've seen a revival of Maori culture. That the setup is not unique to them. One island are sort of upon the same set of claims that the Maoris are here in New Zealand. I mean, which is all times have changed, but in terms of doing science down the road, it's really difficult. So to date, as far as I'm aware, published is still the only papered in Zealand taxa. I sit now, I've learned a lesson, I sit on data for Kunzia. In my title, I fact that we did a phylogenetic study or work on lepidopteran and Kunzia. but I haven't published. I've learned my lesson. I need to get permissions in place first. And it's been 3 years negotiations, and well, I don't know how long. So that for me, to you, is that, you know, Knox, I've been lucky, I guess, in the one hand, being at the right place at the right time, but being proactive as well. Strategy would work in, you know, in South Africa, but certainly here, being proactive, 2 or 3, really benefited the Iberian in that we got funding at short notice without too many questions asked. If you're right place at the right time, there, it would make sense, you know, to make use of that. This whole technique has already been commercialized. You know, in the past, you had to have your own lab, get expensive equipment, get upskilled, and who knows what else. We certainly don't have the time for that or the finances. The commercial labs they provide easy access, skilled personnel, and don't fret about it. It'll probably be in the long. And you know, it's not a one-man show. A team approach assures quality of outcomes when research those you find genomics are complex. So yeah, in this case, I was probably more a project manager than anything else. But yes, I did people in, hence all those. We got the job done in a short space of time. And then finally, the grassroots, regional, national protocols, like in New Zealand, certainly make projects. So keep that in mind, and how the protocols relate are practiced before you you accept, you know, a time constrained project. Make sure that all those permissions before you start. And that ends my presentation. See how long I've talked. Thank you.
For a very interesting talk. I wish if it was not a non-virtual talk, you could have brought us some honey from New Zealand. I think, yeah, you just pushed through your time a little bit, and I think we're now squashed within our time that we might not be able to get a question and answer session. But I also do not see any question in the boxes here. So thank you very much for your talk, which was very interesting, and thank you for availing yourself to the conference and giving us this presentation. We wish you the best in your current work and also your future work. Thank you very much.
Thank you.
So, okay. So colleagues, now we are going to take a short break so that we resume at actually at 10:05, so that we have a bit of time to have a comfort break. From there, we are now going to break into our panel sessions. I think we're going to have 3 like yesterday. The first 2, the one will be on ethnobotany, which will be chaired by Dr. Aida Manduna, while the second one will be on plant physiology and plant biochemistry, which will be chaired by Prof. Sandra Bernard. Then after lunch, we'll actually get into our second panel session again. The first one will again be on ethnobotany, chaired by Prof. Salome Marco. The second one will be on plant physiology and plant biochemistry, and also plant biotechnology, which will be chaired by either Prof. Dave Berger or Prof. Jacques Banner. Then later in the afternoon, we'll get into our last panel session, which will be ethnobotany, chaired by Prof. Lindy McGaw, and the second one chaired, which is on plant taxonomy and plant systematics, which will be chaired by Dr. Mariette Jackson. So I wish you a great day, and I hope you enjoy the activities of today, like what we've been doing for the past 2 days. Thank you very much, and I give back to our organizing committee. Thank you.